and welcome to the latest Genome Giants interview. Today, we are going to be joined by Tak Mak, who is a pioneer in the genetics of immunology. So before we go back and take a look at your career, Tak, if you could just introduce yourself and tell everyone a little bit about what you do as well.
I'm Tak Mak, born and raised in Hong Kong, and parents immigrated to the US when I was a teenager. I went to school and in the US, and then eventually ended up in college, University of Wisconsin in Madison. There I started off as an engineer and didn't really like it that much. And then went into biology, biochemistry, and then eventually I did a PhD in biochemistry. And then went off to work in Toronto with Ernest McCullough, who discovered hematopoietic stem cells. And then after they went back to Wisconsin to work with Howard Temin to learn how to do molecular biology, and started my lab and working on viruses, and somehow serendipitously, we cloned the Tcell receptor genes. And it's kind of like just after you got your driver's license, someone handed you several keys to different cars, and I had no idea what to do. And that's how my career got started.
So if we just kind of take apart like the first kind of things that you said. So let's start off with, you were born in China in 1946, and then you were raised in Hong Kong. What are some of your kind of fondest memories growing up?
Fondest memories. I was a very devoted Catholic. I, in fact, was in the seminary for a few summers to get used to the idea that I would one day be a priest, and then immigrated to the US. And after that, there was I just kind of lost that idea, because I was in a very the environment was not very conducive to continue to be in the kind of religious realms. So rapidly that disappeared, and I became someone that was more out of the church than I was in the church.
You were educated at a religious school growing up. What impact did that kind of have on your education?
Well, first I learned English because they were Irish Jesuits, and became very it's very personal in the sense that I cannot say what would my personality, my outlook of life, and my dealings with other individuals, my empathy, what would it have been if it wasn't for that? But I'm sure it had an impact on how I see the world, and how I see other people in terms of how we all fit into one society.
Did you ever, I know you mentioned that you kind of wanted to go down the route of like being within the church, but did you ever have an interest in science growing up, or did that later develop in your career?
No, I mean, like every kid in Hong Kong, the parents only wanted us to be either doctors or lawyers. Everybody else was a failure. And so when I went to the States, somehow I left. I went to the Midwest, and it was really pretty much on my own. I assumed and kind of took in command of my own life. But my mother, even when I was 35, we already cloned the T-cell receptor. I was full professor. She still asked me, when are you going back to medical school? There is certainly a lot of pressure in that direction.
Yeah. And what were you like growing up as a child? Like, what were your kind of like personality traits, and how has that changed as you've gotten older as well?
How can I don't know. I think I was a very shy person. I had few very close friends. I did a lot of going to church, serve mass. As a hobby, I played soccer. I played marbles. I didn't read a lot of books. I was very bad at Chinese, which I'm very, very regretful, because I think I would like to know more about the Chinese literature and the history, which I think is part of me, and I should be able to indulge in that genetic and environment upgrowing.
Yeah. As you mentioned, you moved to the United States in the mid 1960s. What was this transition like for you at the time?
I had no problem. I really enjoyed it. I went to I went to college in Wisconsin. It was a very so I was supposed to go to Berkeley, and then my mother said, don't go there. There will be a lot of anti-war activities. And so I said, where am I going to go? She said, what other university have accepted you? I mentioned a couple others and said, well, we can't afford that. So go to one that is the cheapest. And that was University of Wisconsin. The irony, of course, was that in Berkeley, the students threatened to bomb the buildings during the anti-war activities, but in Wisconsin they did, right? And there I had a great time. I think the people were really, really nice. They were it's Madison is at that time was a smaller town than now, but it very much integrated into the Midwest mentality. And there were a lot of, before my mother and my sisters moved in, I was there for a couple more years. They looked after me. They treated me very well, and I felt very, very welcome. I didn't feel like I was from Hong Kong and it didn't fit. I right away I felt at home.
Oh, that's really, really nice. Why did you select chemical engineering specifically? And then why did you then later switch your major to biochemistry?
Chemical engineering is, I thought that so remember, every year for the last decades, the university mathematics competition is only one, has only been won by one university in the whole world, and that's Hong Kong University. For some reason, that culture, academic culture, and it just for us to feel to do mathematics is natural. And now I'm not one of the top mathematicians. My classmates, I mean, for 3 of my classmates to be accepted into Caltech, it's, you know, it's really very, very unusual. Right? Even though one actually ended up there, I think one went to Berkeley and the other went to do Chinese literature. To have 3, of course, I did not apply, and I'm sure I wouldn't be accepted. To Caltech, who takes so few students in, and the whole world, 1 high school class, 3 were accepted into Caltech. That says that there is something right or wrong about how we orient ourselves in the mathematics, physics kind of background. And for that reason, I figured I liked chemistry. And my math was not bad, but was not good. So I do chemical engineering. And I was shocked how much I hated it. Because it was not that they wanted to teach you things. It was more like a drill. I remember one time I was given a problem set, and I had a lot of formulas. I had to plug in 100 sets of numbers. And as usual, I go to the bar, I play bridge, and don't manage to actually finish most of my homework. So by the time I did, there was no time. So I did the first 3, and I wrote, if these first 3 are right, I do not understand why I have to do the rest of the 97. I handed it in. I thought, okay, well, let's see what they say. The next thing I knew, I was in front of the dean. I was a woman. Her name was Dean Hoffman. And then she said, well, Mr. Mack, I have a feeling that engineering is not your forte. You could transfer to another university. In other words, we're going to kick you out. Or we could transfer you to another subject or major. So I got transferred to biochemistry. It's with chemistry and biology. I was very happy after that.
What was your time like after that then? Did you much prefer kind of that area of kind of like research and things like that?
Yeah, in the beginning, my first, so this is actually a quite funny story. Our family didn't have a lot of money, and I was given a tuition for basically 1 semester, and I have to make money for my next semester, or I have to go get a scholarship. And having been kicked out of engineering, certainly a scholarship was not eminent. So I went did a lot of things. I did like construction work, and obviously it didn't really work very well because I was 100 pounds. I did gardening. That worked out a little bit, but they don't pay a lot of money. I went dishwashing in the lab of Professor Roland Rickert for $1.25 an hour. And I wash other people's glasswares, and then and finished, and I said, it's only 2 hours. I got $2.50. That's not enough for lunch. Well, actually, it was enough. The lunch was only 80 cents. And so I went back to Professor Rickert, who was a young professor from Berkeley, born and raised in Wisconsin. And he said, well, I have no more glassware for you to wash. And then he said, but if you want to do experiments, I'll pay you $1.50 an hour to do experiments for me. And when you finish, you can wash the dishes that you made dirty for $1.25. So I said, we have a deal. Let's do experiments. And that's how I got started doing experiments. And he was very German. Everything was very precise. I enjoyed the dose response, and I start thinking about, this is how science works.
How do you feel like your time at Wisconsin then kind of shaped the rest of your career going forward?
Well, I mean, from the point of view of getting out of engineering, which actually only took me 3 months to realize that I have no future in, and they don't want me in any case. And I started, I would say, wondering and not really knowing what to do. My professor Roland Rickert, who's still alive, he's retired. He bought himself a farm in northern Wisconsin, and he's gone back to nature. He really looked after me. He made sure that I start thinking. We were in a group called the virology group, and there were some really prominent virologists. There were Paul Caseberg, there was Howard Temin. These are all, and just listening to them say things and thinking what and why they say those things, just made me got more and more interested in what it was all about. Up to today, it's still the main thing that makes me really, really happy is for someone to tell me a piece of results or a connection between two concepts that I had never known. And then it just said, I'm worth it. I mean, I was in Washington, D.C. yesterday, and I talked to Doug Lowy, who's the deputy director of NCI. And he was the one that was instrumental in pushing human papilloma vaccines for young girls first, and then young boys. And we were in a party, and we had a few drinks. And what he told me about human papillomavirus made me felt like that my whole trip down there, just listening to what he said for 10 minutes was worth it, even if I had to walk, because it was very, very amazing. I mean, in essence, what he told me was that there was E6, there was an E7 protein that is part of human papillomavirus, and how they differ between the cervical cancer and the oropharynx cancer. And there were many different dimensions. First of all, cervical cancer is always women. Oropharynx cancer is, for some unknown reason, mostly men. And, was there a sex component to it? Was there a different viral strain difference for HPV? I mean, those kind of things, it's worth. It's worth all
I know
the
what you
time.
mean.
Yeah.
I feel like you learn so much kind of just being in the field, because it's so broad as well, and people have their different specialities, and you're constantly kind of learning from other people, and it's just fascinating.
Yeah.
So you moved to Canada, and you did your doctorate at the University of Alberta. What was that kind of transition like to Canada, and what was your experience like at this university, and how did that kind of compare?
It was not. I moved to Canada to do my PhD, and I didn't get along with my supervisor, and I liked my supervisor in Wisconsin so much, and this guy, his name was John Coulter. He was chairman of the Department of Biochemistry. He had very little time for me, and he felt, because at that time I was ready to start thinking on my own. He wasn't very conducive to the idea that I would tell him that I would rather do these experiments and bring in a new concept. He just wanted me to do this, to do the compared 3 strains of mingoviruses structurally, and I was fine with that. But then I had other ideas of what do these 3 strains do. Yes, they grow different plaque assays, and he had no time for me, partly because he was chairman of the department. And then it was very funny. We made a deal. The deal was, he said, if you get 3 papers accepted by the journal Virology, then I'll give you a PhD, right? So luckily I had an American postdoc, Dennis O'Callaghan, from Mississippi, and he and another young assistant professor, Doug Scraber, kind of took me under their wings, and the 3 of us, we cranked out 6, 7 papers in 2 years, and I couldn't wait to get out of the place.
What was it then like for you? Because as you said, like you had your own ideas. What was it like then being able to kind of set up your own lab? And what kind of challenges did you face during that time as well?
Well, I didn't set my own lab. I went to Toronto, worked with McCullough, and he really changed me. He is several generations Canadian, but he was so British. We would get into a scientific discussion, and all of a sudden, the answer would be a quote from Wordsworth or Shakespeare or Shelley, and that is the kind of, now he's a medical doctor trained for treating leukemias, which in those days was pretty pathetic. You don't have very much except the RSC, which we did a lot of that, and that was great. But I just loved the way he was able to think wide, and it, then later on, going back to Wisconsin with Temin, it was also amazing in the sense that you can think wide, but it has to be very vigorous in terms of experimental kind of like proof. McCullough had more of a philosophical. So it you know, as a quote from a very famous it's kind of like a sailor, and he said, just sail in any direction, you could potentially change the world. And so after we serendipitously cloned the T-cell receptor, all of a sudden the world was looking at this young man, you know, barely 30 years old. And I met Mark Davis yesterday. He was at that time 28. And the two of us had that back-to-back paper in 1984 saying, you know, the hunting of the snark is over. This is it. And there was a News and View written by the late Alan Williams. The title was "The T-cell receptor elusive no more." But then you have these two 30, you know, plus or minus year old, just gotten our position. And then you handed us keys to different cars and different boats. Where am I going to go, right? So that was.
What was that journey like to kind of discovering it? Because I imagine that so much work had gone kind of into it beforehand. There was so much research. And what was it like finally kind of realizing that you had discovered essentially the T-cell receptor?
Well, obviously there is no other word to describe it but luck and serendipity, because I mean, I gave a talk at the my first talk was at Harvard in December 1983, and I was told by the department head that in his department, 16 labs are looking for the T-cell receptor. So who am I to actually even dream of doing it? Now, Mark Davis, on the other hand, he was more committed, and he was more, his journey was more planned and deliberate. So, but beneath the two of us was a technology that very few people had, and those who had were not thinking of the same problem. And that is the technology of hybridization. So as a virologist, the first oncogene that was discovered by Stalin, Varmus, Bishop, and Vogt was in Nature paper in 1976, in which they took the SARC, the Rout sarcoma virus, which has 4 genes, GAG, PO, AMP, and SARC. And then they took the avian leukoma virus, and they had 3 genes, GAG, PO, and AMP. So you notice the one that caused cancer has an extra gene. In those days, there was no cloning. So what they did was they did subtraction of 4 genes and then subtract away 3 genes, right? And ended up with 1 gene. And they discovered oncogene, and Varmus Bishop received the Nobel Prize. So we did it 1 year later, you know, but then we were doing other viruses like fan virus, and with Howard Temin, I did reticular endotheliosis virus. And so that concept that you can find something different. But I wasn't thinking of immunology. Mark Davis learned that technology from Eric Davidson at Caltech, or to do subtractive hybridization, but not for virology. But he was thinking of immunology. Right? So when I come back to Toronto and join the leukemia program, my boss was studying myeloid leukemia. Arthur Axelrod, very prominent scientist, was studying erythroid leukemia. Other scientists were studying B-cell leukemia at the Hospital for Sick Children. That's where most of them were. There was only one leukemia that nobody was studying, was T-cell leukemia. So McCullough said, if you want to join the leukemia program, you study T-cell leukemia. I studied T-cell leukemia. So first thing I wanted to do was to see what genes help T-cell differentiate. So how do you find those genes? 20,000 genes. But I figured, okay, if I take what T cells make and its closest cousin, B cells, and I take T cells, and now you're talking about 5,000 genes subtracting 4,900, right? That's not as simple as that. So in fact, our proposal, when we proposed, was shot down. Some reviewers, foreign reviewers, said, one American said, this is not a project for Canada. Sorry, I'm in the wrong country, right? And the other was, Canadians should not be looking for the T-cell receptor. Moreover, your technology may work for viruses, but will not work for cells because the RNA would be degraded in this hybridization over the 5 days. But you can have phenol. And with phenol in there, the hybridization lasts for a week. So I was end up with no money. McCullough said, I don't care. I will find you the money. So he went to the Hospital for Sick Children and asked Irving Gelfand, can you find TACMAC? And Irving Gelfand said, who's TACMAC? I said, oh, don't worry. Just get the money. We will do the subtraction and we'll see. And when you do the subtraction, there they were. All the T-cell receptor genes, CD4, CD8, CD3, LCK. That was just totally wild.
You've proved them all wrong.
Well, I couldn't believe myself for a long time.
And obviously after that, like loads of like institutions around the world were trying to make you kind of offers, but why did you feel that you wanted to kind of stay where you were in Canada?
I have a personal kind of feeling and belief that I should always remember to thank who for how long. And Canada was a place where it made it happen for me. It was McCullough who believed in me. He went out and got me that $30,000 to do the subtraction, even though the granting agency told me to do something else. I don't want to sound sentimental, but I think that there was to some extent a strong sense of loyalty, and for someone who really went out of his way to help me.
No, that's really, really good. I think that's really, really important as well. Like, as you said, like he obviously did so much to support you. Yeah, and that's really, really nice. You then received support from Amgen to develop the Amgen Research Institute in Toronto. This resulted in your lab pioneering the use of knockout mice. What was this journey like, and how did that kind of differ from your previous kind of journeys?
Because this is a very different world, right? But the person who made it all happen is Larry Sousa. I still play golf with him. Larry Sousa was from Berkeley, then he went to UCLA. I think he is among one of the first 10 people who went to Amgen. Now, if someone asked me, by far the world's most successful biotech company, at least for a long time, now it's a different kind of company. Amgen, it is in many, many, many ways totally, there's no comparison. And there were two people who made Amgen. Me knowing the history. Now, someone who have big egos and thought they did a lot more could send out a assassin to kill me. Then there were Amgen's, like, it went from, it went IPO in 1985 at equivalent today of 8 cents. And the share price today is $250, right? So that's like 4,000x or something. No, like 3,000x. I guess, I don't know, certain companies. But the business was George Rathman, the CEO. And the science was Larry Sousa. And so Larry is a very special person. He's very determined, very quiet. He doesn't talk a lot. Most people hope he doesn't talk a lot, because whenever he talks, it is to tell you you're wrong. And he just, again, one of these situations where like McCullough took on to me, and we met in a meeting, and he offered Amgen to support, and I kept delaying it, procrastinating. It's not that I have anything against taking money to do more research, but eventually he came to Toronto with several other vice presidents and made a deal with my boss, my late boss, Ron Buick, a Scottish guy who plays tennis and golf so well, and absolutely the straightest guy, but really sadly passed away very young, drowned in his cottage lake. So they made a deal to build an institute in Toronto, and then I'll be on loan to go to California once a month for 10 years. And that completely changed my mind of a change in terms of the dimension. It was, like we still today by far have made more knockout mouse than any other lab. I could even say probably even commercial labs, because we had the funding, we had the team, and we had Larry had a vision, and so Amgen's first two drugs is erythropoietin and EPO and Neupogen. Those two, one for red cells, one for white cells. Those two, of course, sales billions and billions. And the third drug was dunisinib, that is homegrown. And that also came from making these animals, and from which you derive the information of how certain genes may work. He had that vision, and he had the influence and the firepower to do that. 20,000 mice.
There's so many.
20,000 different mice. It's not just letting 20,000 mice out there and breed. 20,000 different genetic mice.
I feel like they've had such a big impact on the field, and they've led to obviously several major breakthroughs in immunology and also in cancer. Looking back now and kind of seeing that impact, what are your kind of thoughts on it now, and how does that compare to at the time as well?
Well, science always have a window. That window passes, and then you're on to the next window. So in that time, the window came was why it was capable was sequencing, rapid sequencing, and knockout mice. The two were, but now, I mean, every mouse has been knocked out. Everything has been sequenced. You move on. It's like treating leukemia and lymphoma with CAR T. You're running out. There's only that many antigens on the cell surface, and now you move on. Now you move on to TCR-T, where you treat the patient. When you do immunotherapy, you put in PD-1, PD-L1, CTLA-4. Some T cells unleash themselves and start attacking the tumor. Okay. What are they attacking? Well, I mean, the casual answer is neoantigens that are based on mutated peptides. Well, I'm not a believer of that. So if you take that away, we don't know what the T cells are killing. Because if we do, we can then tailor-made the T cell receptor for your cancer. So, I mean, I was talking to Doug Lowy. I was saying, attacking the virus is a natural, almost 100% cervical cancer is caused by human papillomavirus. And the E7 peptide doesn't change. They get loaded on different HLAs, and you find HLA. And in fact, Christian Hendricks, who is now in New Jersey, treated 11 patients with terminal cervical oropharyngeal carcinoma with a single T cell receptor and he got 11 responses. I mean, so that is checkpoint blockade, of course, is a, undescribably powerful and moved the world. It now becomes the fourth pillar of cancer treatment. Jim Allison, Tasco Honjo, right? I'm good friends with them. In fact, I'm on both of their SABs. But now you say, if you step back and say, if I know, and there's still lots of cancers don't respond at all. Pancreatic cancer, right? Brain tumors. I mean, leukemias and lymphomas do not respond to checkpoint, except Hodgkin lymphoma. And so when you do unleash with a checkpoint blockade removal and the T cells start killing, what are they killing? What is the target? So that would be the next generation of CAR Ts. And that's a lot of work there.
I feel like it's always just so like rapidly evolving the field, and as you said, like it's like the window kind of changes to different things. In more recent years, you have been working like on like exploring like cancer and stuff like that. What are your kind of current lines of research that you're exploring?
Well, I mean, one is of course helping Naoto Hirano, who is this brilliant Japanese professor, came from Harvard, and he was the one who made it possible, because the T-cell receptor, antibody recognizes, an antigen, very high affinity, 10 minus 7, minus 8, minus 9, and even minus 10. And once it grab onto it, T-cell receptor is worse than a cashew kiss. That's it. And if it's right, it stays for 4 seconds. 1, 2, 3, 4. Boom, it fires. And you only need a few T-cell receptors, where for CAR T you need thousands. So Naoto Hirano's lab was able to affinity maturate 51 HLAs, which will cover 90% of the world's population, Asia and Europe. And then with this technology, he will be able to pull out these TCRs. And I think from the therapeutic point of view, that's what he's doing. Targeted genes, therapy, it's also done a lot, but then oncogenes, there are no more new oncogenes, right? So metabolism, so with Lou Cantley and Craig Thompson, we made the drug against IDH, and it's now approved for the treatment of leukemia. It may be approved for cholangiocarcinoma, probably because the phase 3 trial is positive. Gliomas, it would take a while before that gets sorted out. Another was to, cancer cells are genomically unstable, right? They cannot repair all the DNA. That's why they're cancer cells. But if you push them past the point of, if they can repair that much DNA, you give that much DNA, and that's what mostly radiation chemo do. But now you find the genes that actually help them repair the DNA, or prevent them. So that's another category, and collaboration a group of chemists, we have 2 of those drugs in the clinic. And they look like there are some signals. But to answer your question, what intrigues me scientifically, in other words, where is the next direction that I should sail through towards in the hope that may enlarge the world? Is the brain talks to the immune system. Songbirds. You take songbirds, they're male songbirds, they're female songbirds. There's several hundred types, but by and large, if you suppress the immune system of the songbirds, the male songbird suppressed, when they grow up, they cannot sing songs with the same amount of repertoire. So that was actually from last year in March, from Nottingham University, very close to you, was a paper, had a very, very, intriguing title. It says that male songbirds cannot survive on good looks alone. And because if their immune system is suppressed when they were young, they cannot sing songs that are well enough to attract the females. Interestingly, the female birds that were suppressed early can grow up and sing songs. What is the difference? But implying within that is that your immune system and your brain can talk. In 1849, a German anatomist by the name of Rudolf Wagner, he stimulated the nerve in a dog. And then he found something moving. And it was the spleen. Now, crazy enough, 3 years later, it was a surgeon, German surgeon by the name of Henle. And he said, what my friend Dr. Wagner did was a dog. Maybe it's different from a human. And he did it on a decapitated human. I don't know where he found the decapitated human. It was not in the paper, but he showed that the same thing. But that means a nerve that comes, in this case, a vagal nerve comes from the 10th cranial and goes down. And it's a parasympathetic nerve. The nerve, what is in the spleen? What's in the spleen? The immune cells. Anything else? No. And if you look carefully, the spleens, your thymus, your lymph nodes, completely innervated with nerves. Why would nerves need to be in my immune cells? So the genius behind this is a guy named Kevin Tracy, a neurosurgeon in the Feinstein Institute in New York. He collaborated with us about 10 years ago. And I learned all this from him, but we can provide some genetic models to help. And it turned out that T cells and B cells pump out acetylcholine, which is a neurotransmitter. So the nerve tell the B cells and the T cells there is an infection somewhere. And T cells and B cells do what they do. And then nerve will also send back signals to say, now do this, now stop. But it's not that crazy. So of course, there was then this American graduate student I met at the University of Alabama in Birmingham about 8 years ago. I was there to give a talk, and she was the host. And I was able to convince her to come. She deleted the gene from T cells, the gene that makes acetylcholine, which is called acetylcholine transferase. The mouse cannot clear a virus infection. That's it. That's the power of genetics. When we sent in the paper, all three reviewers said the same thing. This is the first genetic proof that acetylcholine is an important part of a T cell function. So serendipity, good friends, amazing people in the lab, 150 of them the last 30, 40 years. And all together, sail in new directions. They may enlarge the world, or they may not, but we will all have fun doing it.
No, that sounds really, really exciting. That's really that's a good path to go down. It's really like kind of exciting research. And as you said, it just kind of shows that the power of genetics as well in that area. I mean, you have made several major breakthroughs. I mean, I haven't been able to list them all, but what has it been like for you kind of experiencing such massive peaks in your career? And how do you kind of deal with situations in your career that don't go so well?
Well, I mean, being a scientist, I think we should be used to failures, and it is part of the game, because we are turning over rocks, and there's not going to be diamonds and jewels or gold or silver or even, copper and iron underneath each rock. So that's part of the game. But once in a while, when you turn over that rock and it reminds you of something that somewhere else, where that rock tells you, and you make that two connections, that's worth everything. That's what it's all for. That's what we were made to do. It's let me tell you a story. 1983, the Paul Getty Museum just got started. I don't know if you ever been to the Paul Getty Museum in Los Angeles. It is a beautiful, beautiful museum. It's on a cliff, Highway 405, driving outside LA. Actually, curiously, it's going west. Everyone thought that there is, LA is there and it cannot go west. literally goes west. And 1983, they decided to get something that is in the museum that would be kind of like the key thing for people to go to see. And they came across a Swiss physician selling a kurus, which is a Greek soldier, a naked Greek soldier. And the story was, it's a 6th century. And they loved it. And so they then sent people out, geologists, chemists, just to make sure, and of course lawyers, to make sure these were all right. All the tests says it is, dates back to the 6th century. It is from which quarry, from which part of Greece. The legal works were all clear. They bought it for just under $10 million, put it in the Getty Museum. And then every now and then, an art expert walks in and says to the board, some of them know the board members, they said, I looked at it, I was repulsed. Another would say, it looks too fresh. Another would say, it's that finger that it should be this, but not that. So it actually started a whole new trend of investigation. It turned out the papers were faked. Okay. It turned out the soil came from Italy. And what is it? These people who walk by, it's that intuition. The one that walk by and say, this looks too fresh, or that I'm repulsed. So they finally took it out. It's no longer sitting there. So I think this intuition thing doesn't come all the time to all people. But I think it's immensely important. Remember also what Richard Feynman said. If you're going nowhere with an old technology, having a fast technology only gets you nowhere faster. I think today, AI, you give a bunch of data to a bioinformatics person, he or she will give you the answer. But is that the answer you want?
It's true. I think intuition is obviously so important in everything you do.
It is.
And how have you kind of maintained such a passion for science? Like, were there any occasions where you thought, oh, I can't be bothered, I'm gonna do something else?
I try to do things for people. You know, I have loved ones who died of cancer. I like to be able to do a little bit more. That is one drive, of course. The other drive is, of course, if you're doing something you like, you don't not have to work another day in your life. And how privileged are we to be paid to enjoy ourselves? It's just, I mean, it comes with the package, which is also failures and frustrations. But at the end of the day, it is what one wants to do.
Yeah. If you could turn your life or your career into a book or a film, what would you name the title? This is a bit of a silly question, but you can take some time to think about it. But what would you call it?
Two Swallows.
I like that. And why would you pick that?
There is a very famous Chinese painter. His name is Wu Kunzhong. And he passed away now. He was trained in Sorbonne in Paris, but a Chinese painter, and he went back to China. He has a painting, big painting, and is a village with a house and some other houses and trees, a river. Very simple. And the painting is called The Two Swallows. And you look at it and say, where's the two swallows? Oh, right here. There is two swallows. So what I took from it, I always wanted to meet him before he passed away. But of course, it's too late. He passed away about 10 years ago, age of 96 or something. I took that as a picture that telling us a message that sometimes in life, and in this case, in his painting, little small things matter. And it will make a difference between those that are a small thing, and if you didn't notice it. Right? I mean, Howard Tennant noticed that brown virus replicate even in the presence of actinomycin D concentration that would prevent a DNA virus from replicating. And since it's an RNA virus, where does the DNA come from? He said, well, there must be an enzyme, takes RNA to DNA, right? Crick, of course, told him he's totally nuts because Crick had the central dogma of DNA to RNA and protein. That's the end. Everybody takes that, write in a textbook, and then keep your mouth shut. Any other crazy person who thinks that protein can go back to RNA, totally nuts. And Tennant said in 1964, in the paper called Virology, and the last line, he said that this may mean there is an enzyme that copies RNA into DNA. And he was being mocked and laughed at. My old boss, Roland Rickett, they're good friends. And he would say, people organize a virology meeting. There's one more spot. Let's invite Howard Tennant. We're just gonna have fun. He was right. that completely changed the world of biotechnology. Can you imagine if you do not have reverse transcriptase? How the heck are you going to make clone genes? No, you can't clone genes without it. And all that from the observation that the concentration of actinomycin D that will allow an RNA virus to replicate, but not a DNA. And that for sarcoma virus, there has to be an enzyme that copies DNA to RNA. And where does the DNA come from? There has to be an enzyme that copies the RNA to DNA.
It's crazy how like he like discovered something and everyone was like thinking he was crazy, but now it's like, for me, like it's such a it's like just it's not even that, like I don't know any different, if that makes sense. Whereas at the time people were like, oh no, like that's not a thing. Whereas now, like that's obviously just an accepted thing. So when you go to school, like that's what you learn about and things like that. So it is crazy.
Yeah, it's crazy.
Thank you so much for joining me today, Tak. I mean, your stories are incredible, and your contributions to the field have been incredible as well. So thank you for sharing.
I'm just so lucky to be part of this big world, sailing a little small boat.
I love that. Thank you so much.
Hello everyone, if you enjoyed this video, then make sure you check out some of our other videos in our series by subscribing below or going to our website frontlinegenomics.com. I hope you enjoy.